Yes, we can see you.
Okay, I'll go ahead and begin. This is about ribozyme reaction mechanisms and the applications in clinical gene therapy for for cancer, brain disorders, and infectious diseases. So we'll start with ribozyme secondary structures. Here's a hammerhead ribozyme secondary structure. You can see helix 1, helix 2, and helix 3, the target RNA molecule from the 5' to the 3' end, as well as the cleavage site. This is a HDV ribozyme secondary structure. Here again, or actually four helices, and then the cleavage site is near the 5' end. This is a hairpin ribozyme secondary structure. You see the helices and the stem loops, and then as well as this classic cleavage site. Here is a scissile phosphate inside the cleavage site of a twister ribozyme. And then here's the idea of a ribozyme vector. So you have ribozyme vector inside a plasmid, and then you have transcription, and then you make the ribozyme. So you have from transcription a ribozyme with a cleavage site and a base pairing element as well. Looking at the cleavage site, this is a hammerhead ribozyme. You can see the 5' has the guide RNA, and then inside you see these ends, these nucleotides, and then this cleavage site. For the HDV ribozyme, it looks a little bit different. Excuse me. You can see the ribozyme cleavage site near the GCs there. So here's the different ribozymes and their cleavage sites. So getting into the mechanism of the RNA self-scission and ligation, it happens by a general acid-base catalysis. So you can see the base attacking the hydroxyl group and then that following the attack to the phosphate. And you make this pentaphosphate intermediate during the cleavage or ligation, and then the electrons delocalize to the downstream base. And that's when you get either the cleavage or the beginning of the ligation. So this is a catalyzed phosphodiester cleavage. So there's three different proposed mechanisms. The cytosine could act as a general base while hydrating the magnesium ion that acts as a general acid. You can see that here in the first one on the left. Or the cysteine can act as the general acid while ionizing the hydrated magnesium ion, which acts as the general base. And then the last proposed mechanism is the chemical model used to investigate the catalytic mechanism of the HDV ribozyme. And you can see the magnesium, the phosphodiester bond, and the base. So, getting to these magnesium ions, the local nucleobase dynamics in catalysis is modulated by the metal binding environments. So, the first ion plays an important structural role by inducing a base pair flip that's necessary in the catalytic fold, which is the cytosine 75 or C75, which moves towards the scissile phosphate in the active site. And then you have ejection of this ion that permits the second ion to bind elsewhere in the active site to facilitate the nucleophile activation. And some quantum measurements and molecular measurement calculations have been done on the magnesium active site of the HDV ribozyme and the role of self cleavage. So it's a general acid role of C75, and the catalysis self cleavage is a sugar phosphate backbone, and the C75 is directly participating. And there's biochemical and structural data that support the general acid role of C75. So in the hybrid quantum mechanical and molecular mechanical calculations, they looked at the reaction mechanism and the changes in the Gibbs energy. So the ribozyme reaction pathway has the N3 protonated C75 proton in the active site as the general acid, and then the partially hydrated magnesium ion that's with one that's deprotonated. So the inner shell coordinated a water molecule to act as the general base. So this supported the notion that the structure of the HDV ribozyme and positioning of the active site of the magnesium ion facilitated a deprotonation and then activation of that 2' hydroxyl nucleophile that I showed earlier. This is some preliminary data that came out in 1998 in October by Jennifer Doudna's group, and this was at 2.3 and 2.4 angstroms. The PDB deposits are here, the 1-DRZ and 4-PR6, as well as the 4-PRF. And you can see the tertiary structure of the HDV ribozyme. It actually weighs 57.96 kilodaltons. It has 2,453 atoms. And it's asymmetric, a C1 asymmetric global symmetry, and it's a monomer as well. They did take another look at this ribozyme in 2014, and they noticed, they used some new tools that gave them another look at the structure, dynamics, and the cleavage. And then I want to get into the ribozyme therapy and the applications in clinical gene therapy. So first we have OZ1 for HIV-1, which was done in phase 2 clinical trial. And then we'll look at the hepatozyme in phase 2, as well as the angiozyme in phase 2 in multiple cancers, but specifically metastatic breast cancer. And then we'll look at an infectious disease, nanoparticles with ribozyme and ribozyme vectors against SARS-CoV-2. And then a hammerhead ribozyme that targets beta secretase, DLST, and BACE in Alzheimer's disease. And then another hammerhead ribozyme that targets ALK or ALK, PTN, VGF, and then FAST-L in glioblastoma. And lastly, we'll look at the hammerhead ribozyme that targets alpha-synuclein in Parkinson's disease. So OZ1 for HIV in phase 2. This was the largest cell-delivered gene therapy conducted trial as of 2009 that supported the concept of OZ1 transduced CD34 cell therapy that is safe and has the most efficacy. There were 74 HIV-1 infected individuals that received TAT-VPR specific anti-HIV ribozyme, which was the OZ1, or a placebo that delivered the autologous CD34 hematopoietic progenitor cells. So this was a major advance in the field. The gene therapy that can be designed to be administered before antiretroviral therapy. So the candidate mechanism for this effect that included OZ1 was the cell-to-cell transmission in the lymphatic system. So there was a compartmentalization of OZ1 containing the cells in peripheral blood, and then the protection of the cell subpopulations, such as the professional antigen-presenting cells or HIV-specific CD4 T lymphocytes. So this is what they did. They infused the OZ1 or the placebo into CD34 positive cells, and then they had a GCSF mobilization with large volume that has the apophysis, and then they selected the CD4 or CD34 positive T cells and cultured the cells and transduced them with the OZ1 or placebo. So it was 30 micrograms per kilogram per day in the GCSF for over 5 days, and the peripheral blood stem cells were collected by large volume apheresis on days 4 and 5, and then again the cells were collected and selected for culture, and then in the presence of a stem cell factor and megakaryocyte growth and differentiation factor, or MGDF, as well as SCF, for about 30 to 36 hours to recruit the cells into cell cycle prep for retroviral transduction with OZ1 or placebo. And then on day 8, the gene-modified cell product was washed and prepared for infusion, and then tested for purity, potency, and sterility prior to diffusion. And then for the hepatozyme phase 2, this was a ribozyme that's prevalent replication of hepatitis C virus. So the phase 2 clinical trial demonstrated a reduction of serum of HCV RNA levels in 10% of the patients on trial. However, there was an animal toxicity issue, and then they stopped the dosing. So the ribozyme was targeting the 5' UTR untranslated region of the HCV RNA genome, which completed the phase 1 and phase 2 clinical trial in patients with chronic hepatitis C. This was done by Merck. The target was HCV IRES near the 5' UTR again, in the 5' UTR, and the disease was hepatitis C virus. For the angiozyme was directed for VEGF-R1, so vascular endothelial growth factor receptor 1, and it had high affinity for the receptor. So this is an anti-angiogenic ribozyme that entered phase 1 trials in late 1998 and the phase 1, phase 2 trials in late 1999, and was put through phase 2 trials in several tumor types. So the serum and plasma levels of VEGF correlated with the prognosis in patients of metastatic breast cancer. So VEGF bound to two receptors in the endothelial cells, VEGFR1 and 2. So this was angiozyme 0, and it was the anti-angiogenic ribozyme targeting the RNA, the mRNA of the VEGFR1. So, although it demonstrated a tolerated safety profile, it lacked the clinical efficacy that precluded this drug from further development. The same company made Herzyme phase 2, and there's a nice little structure of the Herzyme. So, this was being developed in partnership with Elan Corporation in phase 1 trial for patients with HER2 overexpressing breast cancer. So in the phase 1, it overexpressed HER2 cancers, or in patients with HER2 overexpression. This happened at the British Columbia Cancer Agency, and this trial was supported by a business venture of Elan and RPI and Ribozyme Pharmaceuticals, and it was delivered subcutaneously through Elan's Metapad device. So these are two of the products by RPI and Elan, as well as Ribozyme, that are interesting ribozymes that were put through clinical phase trials. Next, I'll move on to the nanoparticles with ribozymes and ribozyme vectors against SARS-CoV-2. This is the latest virus in the pandemic, and is one of the most relevant infectious diseases. So here we have the ribozyme vector, and it's a vector of the ribozyme integrated into it. And then you also have pre-synthesized ribozymes. So you add these to lipid nanoparticles targeted with ACE2. So these lipid nanoparticles have ACE2 on their surface, and then you have a ribozyme conjugated to it. Now here's how it works. So here you have the SARS-CoV-2 virus binding to ACE2 with the RBD and S1 and S2 spike protein, and then it gets internalized into the cell, right, where the genomic RNA is read by a ribosome, and then you have the viral polymerase generating more virus. With the solid lipid nanoparticle, or SLN, you have ACE2 specifically recognized molecules on the outside, and then you have pre-synthesized ribozyme on the inside. And not only the pre-synthesized ribozyme, you have ribozyme vector as well. And this is all in a liposome. So transcription can happen inside the cell. So the nanoparticle enters the cell with the ACE2 specific recognizing molecule, and ribozyme specific gene region or genomic and subgenomic RNA is read. So you have a cleavage reaction with the ribozyme chimera, a DNA RNA hammerhead ribozyme, which you saw the secondary structure of, actually cleaving and making this cleavage reaction, which results in fragmented transcripts. So this halts transcription. So again, the lipid nanoparticles have revolutionized medicine and vaccinology. Here are the latest developments of LNPs by conjugating ribozyme vectors with both pre-synthesized ribozyme and ribozyme vectors using that chimeric DNA RNA hammerhead ribozyme, which would perform the cleavage reaction, resulting in the fragmented transcripts. So again, this halts translation and the viral proteins are never made. So now we'll move to the hammerhead ribozyme, same type of ribozyme that targets beta secretase, DLST, and BASE in Alzheimer's disease. So the RNA cleaving ribozymes, these are molecular tools for silencing beta secretase activity. So it's an excellent target, a therapeutic target or agent specifically for anti-amyloid treatment. These ribozymes control the expression of beta secretase. So this ribozyme efficiently inhibits beta secretase gene expression at both the mRNA level up to 95% and the protein level up to 90% levels. And in the Krebs cycle, you have alpha ketoglutarate dehydrogenase complex that has a subunit enzyme, DLST or dihydrolipoamide succinylate transferase, and this genotype has been studied to contribute to the pathogenesis of Alzheimer's disease. And then with BASE or beta secretase APP cleaving enzyme, that's an attractive target for intervention during the progression of AD. So a hammerhead ribozyme against the BASE gene has been designed and appended with a consecutive transport element on the 3 terminus, and you see a 95% reduction of base mRNA, followed by a 90% reduction of the biosynthesis of base. And then for glioblastoma, you have the same hammerhead ribozyme targeting ALK, PTN, VGF, and FAST-L. So for ALK or anaplastic lymphocyte kinase, this has been studied alongside the hammerhead ribozyme targeting ALK in different expression levels. You have the pleiotrophins or PTN ALK signaling pathways that was probed with the ribozyme expressing xenografts, and that showed the gene dose dependent manner relating to low, medium, and high ALK expression in the implanted cell populations. And these inhibited the pathways that could prove to be effective in treating glioma. So this is a good inhibitor for angiogenesis in vitro in the context of glioblastoma. So these hammerhead ribozymes have been studied in T-cell interaction with gliomas. So HHRZs are targeted to the FAST ligand or FAST-L that help describe the FAST and FAST-L pathways during interleukin 10 or IL-10 biosynthesis after the T-cells confront glioma cells. So interestingly, this IL-10 synthesis by T cells did not pursue apoptosis. So here you see the context. The blood-brain barrier constitutes the parasite, the endothelial cells, astrocytes, and neurons. But you also have the neuroimmunology. So you have the macrophage here and the glioma cell as well as the neutrophil coming into the brain. So you have tumor-associated macrophage, you have IL-1 beta, IL-6, IL-8, TNF, interferon, CXCR1 and CXCR2, CX43, more IL-6, more IL-8, some potassium flux, as well as NLGN3, and then the T cell interacting with the glioma cell, interacting with the TAM cell through CD80, CD86, and CTLA4, as well as PD1 and PDL1 too. So those are common monoclonal antibody ICIs or immune checkpoint inhibitors, but some of those could be toxic. So using a ribozyme like the hammerhead ribozyme is an attractive solution. So the hammerhead ribozyme could be an effective therapy for glioblastoma multiforme. And lastly, I'll go over how the hammerhead ribozyme targets alpha-synuclein in Parkinson's disease. So in Parkinson's disease, you have this fibrillization and aggregation of alpha-synuclein, which is a neurotoxin. And it plays a critical role in neurodegenerative diseases like Parkinson's. So they were able to prevent the nigral dopaminergic cell loss in the substantia nigra by injecting a hammerhead ribozyme mediated inhibition of alpha-synuclein into the MPP treated adult rats. So there was a co-transfection of a adenosine virus with synuclein ribozyme and into the HEC293 cells that resulted in a downregulation of alpha synuclein protein expression in vitro and downregulation of alpha synuclein expression that could potentially be suitable for targeting gene therapy of Parkinson's disease. So I want to thank you for your time, and if you guys have any questions, I'm available for that.